Hello, everyone, and welcome to our webinar. My name is Arabella Kalinei. I'm representing the European CMT Federation today in our webinar. And just a quick introduction, the European CMT Federation, as you already know, is an umbrella organization created to unite all CMT advocacy groups across Europe to help raise awareness and promote better care for people with CMT, Charcot-Marie-Tooth disease, fund research, and encourage the formation of new organizations in countries where these advocacy groups are still not existing. As you already know, in this period, we are running an ambitious awareness campaign in our attempt to raise knowledge about CMT, to educate and to encourage collaboration and to try to spread as much as possible knowledge about CMT, as this is a condition that affects a lot of people in Europe as well as worldwide. So I would like to welcome you to our webinar today. This is our fourth webinar from our awareness campaign. And I'm happy to have with us some amazing experts from NMD Pharma, a biotech leader in neuromuscular disorder research. So welcome Thomas Holm Pedersen, CEO of NMD Pharma. He will dive into science of ClC-1 breakthrough target for improving muscle function in neuromuscular diseases. With a PhD in ion channel biophysics and over 40 publications in top journals like Science, his insights are, of course, unmissable. And we have our second guest from NMD Pharma, Thomas Skerlund Gronebeck, clinical scientist at NMD Pharma. And he will share the clinical journey of NMD 670, a promising treatment for CMT and other neuromuscular conditions. With a PhD in clinical expertise, clinical exercise psychology, and extensive research experience, he brings a valuable perspective to our discussion. So welcome. I'm very happy to have you today with us and looking forward to hear more about your amazing work that you are doing for the CMT community. So the floor is yours.
Okay, so thank you very much, Arabella, for this kind introduction, and thank you everyone for joining today. My name is Thomas Holm Pedersen. I'm CEO of NMD Pharma. I'm also a founder of the company. My background is indeed that I'm a scientist by training. I did my PhD at Aarhus University in Denmark and also spent time at Cambridge University afterwards. And I'm also associate professor at the university, and I still teach medical students. But on the back of the research I was doing in the academic setting, we indeed identified a novel way to improve nerve-muscle communication by inhibiting this particular ion channel that's called ClC-1. I'll tell you more about that later, as a new way to improve muscle functions in a range of neuromuscular conditions. So what we thought we would start out with today is to give you a bit of background on who we are and also where we're coming from. On the science, and I'll cover that part. And then we will actually have Thomas speaking more into the specific details of the particular clinical trial we are conducting now in CMT. The slide you're seeing in front of you right now is, one could say, where it all started. It is actually a photo from a Christmas lunch in a laboratory in a particular university laboratory. We called it the Muscle Laboratory at Aarhus University, where we had a keen interest in working with, indeed, neuromuscular disorders. And you will see that I'm in the back there on the top right, slightly less gray hair than today, quite a few years back. But this was where I did my PhD. And this is actually in this particular lab where I actually also in my PhD proposed that we could use this target, ClC-1, as a new target for treatment of neuromuscular diseases. For those of you who may not be familiar where sort of geographically where we are sort of located, it is showing you on the right there is a picture of Denmark and where the point is in towards where Aarhus is, the second biggest city in the country of Denmark. So if we move on, it was from the discovery in this laboratory that sort of NMD spun out, and we spun out from the university. We formed a company, and today it's grown quite substantially. We're 51 people in the company. This is a particular image from an offsite where the whole company went to offsite to sort of discuss the science, the culture, how we want to mature our company. So you can see it's certainly grown, but we're still a small company, and we are 100% focused on neuromuscular diseases. That's really our focus area. So if you move on, Thomas. The particular focus we have for our company is neuromuscular diseases, and we're driven by, you know, a hope or a vision that we can create a future where patients with neuromuscular diseases are able to live, let's say, better and more independent lives. And we strongly believe a key driver for doing that would be to actually improve muscle function, improve muscle weakness, improve muscle fatigue, find new ways of enhancing endurance of muscle contractions that would facilitate activities during daily life. So our particular mission is that we really want to develop new treatments that can improve muscle function and quality of life of patients with neuromuscular diseases. So that's our background. We came from the academia, we formed a company, and a company with a very specific vision here. So if you move on one more, our pipeline today is very much driven by the original discovery of using ClC-1 as a target for enhancing neuromuscular function. And the pipeline here, which is showing the activity that is going on in the company, and you can see the different lines for the different programs we have. And you can see on top, you can see the different stages the different programs are in. And as you go into the phase 1 and phase 2 and phase 3, that is when the programs becomes clinical stage programs. And you will see here that our lead compound, which is the compound that actually is becoming a tablet and actually is being used to target ClC-1, that NMD 670 compound is now in 3 clinical studies in myasthenia, that is program number 1. It is in spinal muscular atrophy, that is program number 2. And then also in Charcot-Marie-Tooth disease, which is of course the main focus of today, which is program number 8 for us. It is building, actually, these phase 2 studies are building on previous evidence we already obtained in myasthenia gravis patients, where we were able to see that our mechanism is active in man. We were able to see in myasthenia that we could improve muscle function and with administration of our compound. So we're building on already very strong evidence of an active mechanism in man here. If you move one further, the particular compound we're developing and what exactly the product is going to be is actually an oral tablet. It will be taken twice per day with 5 to 8 hours apart. What it does, exactly how it works, is that it enhances muscle activation by strengthening nerve communication with muscle, so nerve-muscle communication, in conditions where there is failing transmission of electrical signals from the nerve to the muscle fibers, and therefore the muscle fibers are not activated. But with this approach, we can actually enhance that nerve-muscle communication. And what we are hoping to see here is that we can improve muscle strength, we can improve power, we can improve endurance, and that we hope will translate into a better, let's say, mobility, a better ability to sort of handle everyday life, and a better ability to actually conduct a number of daily activities. So if we move on, a little bit more specifically on what it does. So I mentioned that by inhibiting this particular ion channel with NMD 670 or other ClC-1 inhibitor compound, you can enhance the nerve-muscle communication. And this picture tries to exactly describe that in detail. You will see here a very simplified version of what we call the motor system, including, of course, the brain, the spinal cord, the nerve that connects the spinal cord to the muscle, and also the point where the nerve is actually attaching to the muscle fibers or the muscle cells. And the key here is that the signals to activate muscle fibers are these blue lightnings you can see here, and they have to travel from the brain down the spinal cord into the peripheral nerve that goes over to the muscle, and there the blue signal has to travel into the muscle and actually conduct along the muscle fiber length in order for the muscle fibers to contract. And unfortunately, there are a number of conditions where the last point in this, the signal transmission of the blue light or the electrical signal from the nerve to the muscle fiber fails, and therefore the muscle fibers are really never activated appropriately. They are, so to speak, not told to be activated. And what we have discovered is with our molecules, if we inhibit ClC-1, this particular chloride ion channel, which you can see in the highlighted circle, by inhibiting here, we can actually boost that transmission of the nerve signal to the muscle and thereby restore the signal traveling into the muscle fiber and restore the ability to activate the muscle fibers. And as highlighted here by this publication here on the right, this is something that we have already taken through all of the early development into patients and actually demonstrated that it actually is active and works in patients with myasthenia gravis. And if you go one further, Thomas. We are also now actually able to show that, and this is a paper that will be coming out very shortly, that we also have a similar ability with our compound to restore neuromuscular signaling or the nerve-muscle communication with NMD 670 in animal models of Charcot-Marie-Tooth disease. So let me perhaps dive a little bit more into why we hope that we can do something. What's the rationale for Charcot-Marie-Tooth disease specifically? We started out by actually exploring whether there is a deficit between the nerve and muscle communication also in Charcot-Marie-Tooth disease. And without going into a lot of details, what we essentially measure are electrical signal transmission from the nerve to the muscle in patients with Charcot-Marie-Tooth. And if we have events, what is known as blocking, that shows that the nerve signal is not able to get into muscle fibers. So if there's block, it means that there is failing transmission between the nerve and the muscle fibers. And as you can see from this graph here, compared to healthy control, we did observe that in Charcot-Marie-Tooth disease, there was substantial blocking between the nerve and the muscle. And that, of course, is potentially an important contributor to the weakness and fatigue that is experienced and associated with this disease. So what you see here in the middle panel, or in the panel 2, is exactly that notion, that if there is worse nerve-muscle communication, well, then you have a worsening of the muscle symptoms. So if the nerve cannot activate the muscle fibers, then you will have the muscle symptomatology. And that is exactly what we know that our new approach can actually restore. We can basically improve nerve-muscle communication, and we hope that will translate into improvement also of the muscle symptoms. So if you click one more, we tested that in CMT animal models. And what you are seeing here is the effect of one of our compounds, actually NMD 670 specifically. And if you look at the blue bars, you can see that they're pointing upwards. And these were the CMT animals that were actually administered our NMD 670. And this is the recovery of contractile force. So you can see with different intensities of contractions, this is what is known as frequencies of stimulation. You can either have light, more intense, and then intense contractions. In all situations, we actually saw an improvement of the muscle strength with when administering NMD 670 to the animal models. However, if they were administered in the wild type animals, we are not seeing any recovery. So we know that our compound in animal models can improve muscle function. We know that what is actually being recovered is the nerve muscle communication in the animal models, and we have identified that also in Charcot-Marie-Tooth, for the first time actually, that also in Charcot-Marie-Tooth there is deficits between the nerve and the muscle in the communication. So I think this is the rationale for moving forward with this new approach that we hope will improve muscle functions in CMT. So from there, I'll hand it over to Thomas to take us through exactly what we're doing in the clinic. Thank you.
Thank you so much, Thomas, and thank you, Arabella, for the opportunity to address the CMT community. Fantastic opportunity for us. As mentioned in the beginning, my name is Thomas Grønnebæk, and I work as a clinical scientist for NMD Pharma, and I have been with NMD for 4 years now, since we actually started the clinical development program with this particular molecule. Let's dig a little bit deeper into what we are now doing in the CMT space with NMD 670. So based on all the preclinical and clinical data that Thomas just showed, we have designed a phase 2A study to investigate the safety and efficacy of NMD 670 in ambulatory adults with CMT types 1 and 2. The objective of the study obviously is to, for the first time, investigate if this mechanism also works in humans with CMT. And in order to do so, we have designed a randomized, controlled, parallel group study in which 80 CMT participants will be randomized to receive either placebo twice daily or NMD 670 400 milligram twice daily for a total of 21 days. Before going into the actual treatment period, there will be a screening period. The screening period is up to 28 days. And then after the actual treatment period of 21 days, there will be a follow-up period of 7 days. There are some particular design elements of the study that I want to highlight to you. And one of them is that we plan to enrich this study with approximately 30% of the participants that have CMT type 2. And the reason for this is that we want to have sufficient number of participants to explore if the mechanism works better, equally well, or maybe not so good in CMT type 2 versus CMT type 1. The primary endpoint of the study is the 6-minute walk test, where participants will be walking for 6 minutes, and we will then measure how far the patient is able to travel over those 6 minutes. But we include a range of different efficacy endpoints to ascertain muscle function across different muscles of the body, and also to ascertain different components or different elements of muscle function, ranging from muscle strength to fatigue and balance. And then, of course, we also include patient-reported outcome measures to really ascertain if the mechanism is also able to improve quality of life and fatigability. Some other key elements of this study is that the study duration will be up to 8 weeks, and it will include 6 on-site visits. There will be administration of NMD 670 for a total of 21 days, and those doses will be taken twice daily as an oral tablet at home. And during the study, you can continue to receive the usual care, meaning if you're taking pain medication or if you're receiving physical therapy, it's completely fine to continue with these therapies. As this is a randomized trial, some patients will be receiving NMD 670 and some will be receiving placebo in the study. In this study, we will randomize participants 1-to-1, meaning that there is a 50% chance that you will be receiving NMD 670 if you participate in this trial. Because this is an early phase clinical trial, we have some quite strict inclusion criteria and exclusion criteria. And the purpose or the meaning of this is to make sure that we have a rather homogeneous population, which will really increase the likelihood of a positive study, which will allow us to continue and expand the development program in CMT to more CMT types and to participants that are not able to immediately participate in this study because of the inclusion-exclusion criteria. Nonetheless, let's go through some of the most important inclusion and exclusion criteria. In order to participate in this study, you must be between 18 and up to 70 years of age. And then you will need to have genetically confirmed CMT type 1 or CMT type 2. There are no restrictions with regards to subtype of CMT1 or CMT2 as long as we have a genetic confirmation of the disease. We also require you to have a certain level of functional ability. You will need to be able to walk within 100 meters of age and gender adjusted normative value from the 6-minute walk test. And the objective of this particular inclusion criteria is to make sure that we again include a relatively homogeneous population, that we reduce the variability and thereby increase the likelihood of seeing a positive signal at the exit. Then we also require you to have mild to severe ankle weakness. So we know that there is some room for improvement. We require participants to have a body mass index of between 18 and up to 35 and a minimal weight of 40 kilos. And then we have some contraceptive requirements for the study. Because this is an early phase study, we want to make absolutely sure that we protect you as much as we can during the study. And therefore, you are required to use contraception in accordance with the local regulation. We have a number of key exclusion criteria also, and the first one is that any other significant disease that may interfere with the objective of the trial will be exclusive. And then severe deformity or ankle contracture that, in the opinion of the investigator, would impact performance on the different endpoints is also exclusionary. And then we are excluding participants that are using ankle foot orthosis or other assistive devices. The reason, again, for this is to get a relatively homogeneous population. We do think that we can also improve muscle function in participants that are using AFOs, but we really want to make sure that the study is successful, and we believe that we have the best chances if we get a relatively homogeneous population in this initial early stage study. Then we are also excluding female participants who are pregnant or who plan to become pregnant or are currently breastfeeding. And that is again because of the early phase nature of the study. We want to make sure that we protect participants as much as possible here. The study is already up and running. We have actually recruited 5 subjects now across the US and EU. This slide here is showing you a list of sites that are up and running and will be up and running relatively shortly. As you can see, we have 2 sites in Belgium, 2 sites in Denmark, 5 sites in France, and 2 sites in Spain. We would have loved to welcome more countries across the EU and other regions, of course, but this is a relatively small study, and therefore only a smaller number of trial sites are actually needed to deliver the 80 participants that we are looking for here. We have a number of sites already recruiting, and I will come back to how you can get in contact with each of these sites in case you're interested in participating in the trial, or in case you know somebody who might be interested to participate in the trial. This is showing you a comparable list of US sites in case we have any US or North American attendees. There we have 3 actively recruiting sites at the moment, one in Columbus, Ohio, one in Columbia, Missouri, and then one in Austin, Texas. If you want to know a little bit more about the study, if you want to know the status of the individual trial sites, you can always visit this webpage, which is called clinicaltrials.gov. You can use the NCT number, which is written here, or you can use the QR code, which will take you directly to this webpage. And there you will see the list of sites. You will see which sites are actively recruiting or not. And then you will also be able to see contact information for the different trial sites, so you can reach directly out to these sites. If you have any questions, or if you cannot get in contact with the sites using the published contact information, you're more than welcome to reach out to me directly, and then we are happy to facilitate the contact for you. And with that, I just want to extend acknowledgement and appreciation to all the individuals and families who participated in the trials that we presented. Of course, also to the investigators and the site staff that are performing this ongoing study. And then a big thanks to all of you for listening. Thank you so much.
Thank you so much, dear guests and the experts in the CMT field. It is amazing to see and very encouraging. After all, it is really encouraging to see that there is hope out there for CMT patients, and we are so happy to see there is research going on, and hopefully we'll have something to lower a little bit this burden that this disease causes in our community. So I would encourage people to ask their questions, as I've seen we already received some questions, and I will start from the chat. Yeah, so Thomas is asking, did I understand correctly, you expect a significant improvement after 21 days?
So a couple of things here. We certainly expect to see positive improvements in muscle function over the course of 21 days. In fact, in the previous study that we already conducted in patients with myasthenia gravis, we were able to see improvements, statistically significant and clinically meaningful improvements in muscle function over the course of hours. So this is an acutely working mechanism, and we should be able to detect immediate improvements in muscle function. So therefore, we also expect that the duration of 21 days will be sufficient to see positive improvements in muscle function in CMT population. Very good question, by the way.
Yeah, it is really promising. And Alexandra, our board member from European CMT Federation and president of CMT France, first of all, he's thanking you for such a brilliant presentation, and he said, glad to be involved at this level in France. If you need any more help for recruiting patients, we would be glad to help you. So he's offering his help with patients in France.
Yeah,
like every CMTF members, of course. We have this good partnership, and thank you for supporting our awareness campaign, by the way.
Yes.
Oh, that's fantastic news. I'm sure we would love additional help with the recruitment, and we'll go back to our medical colleagues and discuss how we can facilitate that, and that goes for all of you, of course. Thank you so much.
Thank you. So Larissa then is saying, thank you for the presentation. You showed a graph on CMT2D animal model. Do you have data on the drug beneficial effect in animal models other than CMT2D? While analyzing the trial results, will you look into separate subtypes of CMT1A, 2A, etc., or only in CMT1 and CMT2 versus placebo?
So on the preclinical, we also have data on a CMT1A mouse where we saw similar effect to what we're seeing actually on what we showed. So we have clinical rationale from CMT1 and CMT2, given what we conducted in the clinical observational study, where we looked for nerve-muscle communication deficits. And then on the preclinical side, we have corresponding data also in CMT1 and CMT2, showing that there we have efficacy with the treatment approach. So we do have support for those.
Okay, great. And Patrick is asking if you have a pediatric plan in place. Are you assessing a possible oral liquid formulation for children?
These are great questions. Well, first of all, we are of course fully aware that there will be also a pediatric. It could well be relevant also for pediatric patients, and it is part, of course, of the planning going forward. And I think maybe at a very high level, we just want to also make clear that all of the inclusion and exclusion criteria Thomas said before, it is really in the interest of making sure that this can move forward. We really want to build the best opportunity for us to show that the mechanism is active, so we can actually move forward to a broader population of CMT, including the pediatric. So there is a pediatric plan that's being worked up at the moment. It is something that also is required from the authorities. So we have a pediatric plan for an approach like this. So yes, it's coming. It does require some additional work, both in the clinic, but also in the non-clinical areas that we are doing.
This is great because we do have children and we are hoping for something for them as well.
That would be wonderful. So do we. So
yeah. And Jeppe is asking, do you have some insights on the acute versus longer term effects of intervention? Example from other trials.
Yeah, no, this is great question as well. Really good question. So we are indeed seeing the rapid response that we are both seeing that in the animal models, but also in the clinic, and that rapid response is reflecting that the nerve and the muscles are better able to communicate, and hence you can actually get stronger muscle. What we have observed, and I have to stress that this is only observed in animal models so far, is that with repeated administration, and 21 days in patients probably will not be enough, but we don't know yet. But with repeated activation, what we've seen in animal models is there is a second phase of improvement where you actually have after the sort of acute effect, you're seeing a longer term improvement also in the nerve and muscle connection. So this is in animals. So we believe very much at this stage that this is in part driven by the fact that there is a better communication between nerve and muscle, so you can reactivate the muscle fibers. So you're actually able to stimulate the muscle fibers to grow, stimulate the nerve to grow, stimulate the whole interaction in the motor system. And I think it probably appeals to almost an intuitive sort of understanding of it, because we all know that we need to be active and train in order to keep our motor system working for instance, as we get older. And also conversely, if we are actually not able to move, if one becomes very inactive and bedridden, you're losing the system. So by what we're doing here, bringing back the nerve-muscle communication, we over time are seeing a sort of gradual further increase in function. So far only in animal models, but of course this would be wonderful if we could have that sort of second recovery phase, and also see that in human. Too early to say for sure yet.
And still a question of my own, just to link with what you've just said. When it comes to patients with severe foot deformities or patients who had surgeries, reconstructive surgeries, for example, do you expect that they will benefit from the treatment as well, or will they see some improvements?
Yeah, it's still early days on all of this. So this let me just also stress that this would be at this stage a little bit speculative, and the ongoing study will not be able to address your question. Just to sort of set that. But what we have seen in animal models is if we actually cut a nerve and wait for it to grow back to a muscle fiber, grow back to the muscle, sort of re-innovating, reconnecting, essentially, that is something that we can see being facilitated with this approach. And it comes back to the same rationale that if there is a connection, as soon as it starts to connect, that connection will be stronger if we're treating with this approach. And that in turn stimulates basically a growth signal so that the connection actually grows back better. But I really just want to emphasize that this is early.
Yeah, okay. And we do have a question from our Vice President, Ingolf. Yeah, so do you expect side effects? What about muscles? What about the nerves? So he's more asking about possible side effects.
Yeah. ClC-1, as to a starting point, ClC-1 is only expressed in skeletal muscle. So sometimes this is very often a challenge when you're developing various new therapies, is that maybe the target you're approaching, the protein you basically want to focus on, is not only expressed in the tissue where it's relevant. Sometimes you have the target being expressed in different areas, and that can cause side effects. However, in our case, ClC-1 is only expressed in skeletal muscle, and that of course is a major benefit. So it in that sense it's very tissue specific, and that sort of advocates in favor of a very sort of focused side effect profile if there is any. And Thomas, perhaps you can also speak to what we are sort of also making aware and patients being aware of as we go into a study, but the underlying biology here is such that if you were to inhibit the channel excessively, and if you were taking too much of ClC-1 inhibition, you could get some stiffness of the muscle. But Thomas, do you want to say exactly what we also communicate in connection with the trial on this topic?
Yeah, absolutely, Thomas. So maybe first to say that before doing this study, we have already conducted a study, a phase 1 study in 57 healthy volunteers and 12 patients with myasthenia gravis. We have also exposed approximately 30 patients with spinal muscular atrophy to this drug, and no patients so far have developed severe or serious side effects. So that is the first thing I want to highlight. Then the second thing is that this is still early days, so we are monitoring very, very intensely for any side effects that could be associated to the drug. So far, we have not seen anything aside from what Thomas alluded to earlier, that when you give this drug at very, very high dose levels, much higher than those we are testing in the current study, we can see muscle stiffness, also called myotonia. Then we have seen a number of other side effects which have been similarly observed in participants receiving placebo, and therefore, because it's early days, it's very difficult to say if these are related to NMD 670 or not. So far, it doesn't look that way. But because it is an early phase study, we monitor for side effects very intensively during the study to make sure that all participants are safe.
Yeah, this is good. Okay, we do have another question from Anne Fisher. Can CMTX CMTX patients also participate in the study?
That's a very, very good question. Unfortunately, the answer is no. At this stage, we are only enrolling participants with CMT type 1 and CMT type 2. we do very much believe that the mechanism would also work in CMT-X. We have no reason to think not, but because the preclinical studies and the observational studies did not include CMT-X type participants, we, again, for the reasons stressed in the beginning, wanted to increase the likelihood of a positive study and therefore initially focused on CMT types 1 and 2. If the study is successful, we will also roll this out, of course, to other types of the disease, including CMT-X.
Okay, thank you. Filippo, you wanted to?
Yeah, just to be 100% sure, when you say CMT1 and 2, you mean 1A and 2A?
We mean all subtypes of CMT type 1 and CMT type 2.
Yeah,
but
I'm
not
asking this
X-linked,
because
but not
yeah,
X-linked
okay,
CMT
that's
types.
Got it.
Yeah.
Thank you.
And Rachel, I guess she's from UK. She's asking, did you say you had any UK participation possibly?
We don't have any UK trial sites in the current study, but if you are willing and able to travel to some of the trial sites across Europe, that is a possibility. We have arranged for the possibility to cover travel expenses, so it is certainly a possibility. If it's feasible to you, then you're more than welcome to reach out to one of the trial sites or to me, and we can help you facilitate contact.
Okay. And Ulla Britt is asking, do you have any ideas for people having clear CMT symptoms but still without genetically proved status? Would there be any risk for them? Yeah, there may still be patients who are not genetically tested, but all signs are there. So,
what I will say is that in order to participate in this study, we need a genetic confirmation of the disease. We can offer one-off, I will say, genetic testing under the protocol in case you do not have a genetic confirmation of the disease. Of course, there are people out there living with CMT symptoms where CMT is suspected, but where the subtype have not yet been identified. And of course, the only thing that we can really hope for and also support is more research into finding these subtypes, more genetic research into finding these subtypes. We certainly don't expect that there would be any risks in participating in the study, but unfortunately, it's not possible without a genetic confirmation of the disease. I hope that answered the question.
yeah, and someone from Germany is saying that if you are still looking for persons, can German take part, and which would be the way to be part of this clinical trial?
We're absolutely still looking for participants. I think so far we have dosed or included 5 out of 80 participants. So there's much room still for participation. The trial will run, we expect, at least for another year. So there should be plenty of opportunity to participate. German participants are more than welcome, as are any other candidates from across the Europe and other regions. I think the most easy route for participation, if you're based in Germany, would be to go either to Belgium or Denmark or France, dependent on where in Germany you are located. So please reach out to me afterwards, or please have a look at clinicaltrials.gov and identify the site that is closest to you, and then feel free to reach directly out.
So basically what you're saying is that you have to be present and go to a center, because Peter is asking, can you participate remotely? Is it enough to visit on day 1 and day ClC-1, but I guess this is not an option, right?
Not in the current study, unfortunately. 6 on-site visits are required. This is to obviously do the functional tests to see if the drug works, but it is also to monitor the safety of the drug. So we do require in-person visits 6 times over 8 weeks. In the future, in larger studies, it certainly could be a possibility, but not in this first inpatient study.
Yeah, we do have here a question from Rachel. I'm not sure I completely understand it, but I think she's trying to ask what is the time frame to possible pill production from efficacy. I'm not sure after the trial is ended, or I'm not sure exactly. Yeah, I think time frame to.
So what the usual approach here is that, first of all, the study we're doing right now is assessing whether the mechanism is active in CMT. Then subsequent to that, we would have to run at least one more larger, substantially larger trial. And that probably would suffice for registration beyond that point. What we're looking at here is probably 2 years, 2.5 years at least, after this first trial has actually completed. So there may be ways of actually accelerating this, particularly in CMT, because as we all understand, this is an area where there is high unmet medical need, and there is also less treatment options. So of course, this is a discussion to be had with the regulatory authorities about how fast we can move this. But we will definitely try as fast as we can. But we are talking at least one large study after the current study.
Jeppe is asking, do you measure actual differences in their daily life activities during interventions? For example, using accelerometers before, during, and after, or only the pre versus post measurements?
It is something that we are looking to incorporate into our trials. It is not currently included in either of the protocols. And the reason for this is that up until very recently, there have been no good qualified endpoints accepted by the regulators. This has changed recently with qualified endpoints using accelerometers in Duchenne, which is now also being rolled out in other neuromuscular diseases. So it is something that we are super interested in and that we would love to include into our trials. Whether it's going to be this trial or a future trial, we cannot say for sure, but it's certainly something that we're interested in.
Okay. And Roger here is having a nice comment on the question, speaking in electrical terms. So you are trying to improve the connector of a cable, but not the cable itself. In CMT, either the cable insulation or the central conductor is inhibited or damaged. Will NMD 670 be able to help here as well?
Well, I think it's a very nice way of describing it, in fact. Sort of at a first sort of instance, it's kind of amplifying the available signal, to sort of stay in the electrical terms. It's amplifying the signal that is diminished, and thereby it becomes large enough to actually make the signal travel along the muscle fiber length. More biologically, what that can actually do over time is when the nerve and muscle is interacting, we do believe that once it's being used, it will actually also grow and recover, better. But you're absolutely right. It's not on the cable actually changing the insulation per se. It is rather boosting the magnitude of the signal that is available between the nerve and the muscle.
Okay, and then we have there a question again about side effects. You kind of already said about them. Are there any other side effects that you observed in animals, for example, besides those that you already mentioned?
The dose-limiting observation in both animals and actually also in our phase 1 that Thomas described before has actually been the on-target pharmacology. So but at much higher doses than what we're actually using in this study. So that's really been the dose-limiting observation.
In previous clinical trials with NMD670, as I also alluded to before, we have also seen a number of side effects, such as headaches or fatigue, but these were equally seen in patients treated with placebo, and therefore we don't at this stage think that it is related to the study drug, but because it is early phase clinical research, it is still too early to say. And it is something that we are monitoring closely for.
Okay. And Alex is asking, when do you expect to publish or communicate the results of this phase 2 clinical trial phase?
So naturally, we will have to complete the study first. We are still sort of as we are seeing the first participant being randomized, and we're sort of getting a good sense of how fast the study is progressing. We will get more clarity on that. But clearly, we have to complete the study first, analyze the data, and then publish it. Meanwhile, I do want to just stress that we will have a paper out which explains the clinical observation study we did, which we call the established study, and then also our preclinical data, and that has already been accepted and is actually already in process for being published. So this may well be available online. I would say certainly within the next quarter, but again, not in patients with this compound, but in preclinical models.
Okay. And Horma is asking, do participants have to stay near the university, so basically near the clinical trial sites for 28 days of the study? Or maybe a better question would be, how often do they need to go for this regular evaluation?
So there'll be 6 on-site visits over the course of approximately 8 weeks. I think the best thing to do here is to reach out to the trial site nearest to you and then figure out what the logistics around the participation will be for the individual, because it will also depend a little bit on the trial site. So that would be my recommendation.
Okay, and Tiziana is asking, should you subdivide patient analysis affected by CMT1 and CMT2 according to the gene involved?
Yeah, absolutely. We will do many different types of subgroup analysis to understand if the drug works equally well across types of CMT, across different ages, across different baseline characteristics, such as how severe the disease is at baseline. All this will be analyzed in great detail to really understand who can benefit from this drug.
Yeah, and
but the main analysis will include the entire population.
Okay, so Thomas here is saying, could this drug also be used in the field of sports to treat injuries or even as doping? This could make financial much easier.
Well, it's interesting. We often get the question about doping, actually, and we do not believe it has any application at all within that sort of in that area. And the rationale is that if there is a good connection already between the nerve and the muscle, you cannot make that better. If it's already working, there's really you can't really make that stronger. So it's like turning on the light. So if you've turned on the light, it's turned on. But if the bulb is not really working well, then you can maybe do something to fix it. So in athletes, you naturally will have a good connection between nerve and muscle, and so they will not benefit from this. And I think it's very important that we don't have that potential misuse of this.
Okay, we do have a question here in Italian, so if you can translate, Filippo, I kind of understood.
Sure. Yeah, that's basically a patient, Pietro, who's asking whether CMT2 related to EGR2 gene could be also included in this study.
Sorry, can you repeat that one more time, Filippo?
Sure. It's CMT2 form related to gene EGR2.
Yeah, absolutely. As long as you have genetic confirmation of the disease, and as long as you have a subtype of either CMT1 or CMT2, you are potentially eligible for the study.
Okay. And Alex is asking, what do you think about including MRI as biomarker for a longitudinal study? Many studies are showing it could be a reliable one.
For a future study, this is something that we would be considering as well. For the current study, we believe that it is not really long enough dosing to pick up the changes that you need to see on an MRI. But with a longer dosing, it's part of options we have going forward. But it's definitely an emerging one, which is also on our list.
Okay, and just very quickly, please, this is a personal question. What about patients that have both CMT and other neurological conditions? Like, for example, my son has epilepsy, or I don't know, there are patients with MS and CMT even so. Do you think this future treatment could be contraindicated for them? I mean,
the only neuromuscular disease I can think of where we probably would not want to give this is myotonic diseases, where you see too much muscle contraction. But I think it will be extremely unlikely to have CMT alongside a myotonic condition. In the case of this particular trial, we do have an exclusion criteria which is around other significant diseases that could affect the study outcome measures, and it cannot be excluded that some of the diseases that you just mentioned would also be included on such a list. And the best thing to do here is to go to one of the sites to talk to the investigators, and then they will be able to tell you if you are eligible or not.
Okay, thank you. Filippo, we have another question here in Italian, but I'm not sure.
Yeah, there's first Herma asking the code for further information. Maybe she refers to the clinicaltrial.gov websites. So if you please, Thomas, can put it in the chat, so she will have it. Thank you. Yeah,
we can also share the presentation with you, Filippo, and Arabella, and then you can distribute it to.
That'd be great. The webinar
the
as well will be put on our YouTube channel, so it's not a big deal. So yes, Pietro is asking whether. Well, we also spoke about the timing of the clinical trial. It was just asking whether patients suffering from kidney issues might be also included into the study.
I think again, you would have to reach out to the investigators. They will then help you determine if your other condition makes you eligible or not for the study.
Yeah. So the Ingolf's last question, Arabella, and then we are.
Yeah. Yeah. So Ingolf is asking, how do you find and select the centers to participate in the trial phase 3? No German was selected for phase 2. What is the reason? Yeah. So why you did not select patients from Germany as well?
So when we started to embark on this process of doing a phase 2 study in CMT, the first process is called site feasibility. This is where we identified potential sites for the study, and I'll say we did not decide against Germany. We just don't need that many sites in this initial study to deliver 80 participants. And we did already have collaborations with some of the other centers, and therefore these were chosen over German centers. I will say that in our study in SMA, we have included Germany, And most likely, I'll say very likely, if a phase 3 study was to be conducted, it would also include Germany and other countries, of course, because many more patients would be needed for a phase 3 study. So many more centers would also be needed. The criteria for the centers are, of course, that they have the capabilities to conduct the clinical trial, that they have the know-how and expertise to do the assessments that we are performing, that they see enough potential participants to make it worthwhile to set them up as a clinical center, because a lot of work goes into setting up a clinical trial site for participation. It takes a long time, both for the site and for the staff running the study.
Okay. So as we reach the end of our webinar, I want to thank both of you for your insightful presentation and for bringing so much information, and not only information, but also a little bit of hope for our CMT community. Thank you for your hard work and for accepting to participate in our webinars. Thank you all for joining our webinar today. And please don't forget that this webinar is recorded, and you will find it later on our YouTube channel. Don't forget to follow our activity, especially now during our awareness campaign. Follow us on social media, Facebook, Instagram, LinkedIn, and of course on YouTube. And don't forget to join us on December 7, starting with 11 a.m. CET, for our 5 exclusive webinar with CMT expert Dr. Vincent Timmerman. We'll share groundbreaking insights from his decades of research on CMT. So thank you all for joining us today. Do have a wonderful weekend, and see you soon with our next webinar. Bye-bye.